Welcome back to Parasitology. Hi, I'm Christopher. In this video, we are looking at two groups of insect ectoparasites that are small enough to miss and important enough to ruin everyone's day: fleas and lice. Both belong in class Insecta, but they use very different strategies. Fleas are jumping, blood-sucking insects with a life cycle that partially unfolds in the environment. Lice are host-specific clingers that spend their whole life on the animal. Learn the pattern: what the parasite looks like, where it lives, how it feeds, which host it prefers, and what clinical clues it leaves behind. If you sort those patterns, the species names start behaving more like useful clues instead of Latin confetti. Let's start with the big split. Fleas and lice are both wingless insects, but they lead very different lives. Fleas belong to the order Siphonoptera, and the name is helpful. Siphon means tube or pipe, and aptera means wingless. So the name points us towards a wingless insect with sucking mouth parts. That's almost too useful. Every once in a while, taxonomy throws us a snack. Lice belong to the order Pithoraptera. Now, the word kind of looks like someone sneezed into a spelling test, but it points us towards lice and winglessness. Lice are flattened, clawed, host-associated insects that are built to cling on. Remember, for dog and cat fleas, especially Tennisipalides, adults remain on suitable hosts once it's established. Eggs fall off, and the larva and pupa develop in the environment. Lice? Very different. Eggs, nymphs, and adults often stay on the host. Fleas are a host plus environmental problem. Lice are mostly a host and close contact problem. Tiny insects, very different life strategy. Now let's zoom in on fleas. The word Tennisipalides is a gem. Tenno means comb, and cephal points to the head. These are comb-headed fleas, which fits because Tennisipalides species have combs that help us identify them. The big one in dogs and cats is Tennisipalides felis, the cat flea. Do not let the common name fool you. The cat flea is commonly found on both dogs and cats. Apparently, it just didn't read the label. Tennisipalides canis, the dog flea, it exists and matters, but in many settings is less common than Tennisipalides felis. Pulex irritans, the human flea, has a broader host range and public health impact. Echidnophaga gallinaceae, the stick-type flea, is very memorable because adults attach firmly, often around glabrous areas of the head of birds and other hosts. The name Xenopsilla chiopus is oddly memorable. Xeno means foreign or strange, Scylla means flea, and Chiopus points us to the Great Pyramid of Giza. So the oriental rat flea becomes the foreign flea of the pyramids, a sticky reminder for flea and murine typhus. The big clinical idea is this: flea species and flea ecology matter. For the dog and cat fleas, treat the animal and the environment. For rodent-associated fleas such as Xenopsilla chiopus, host switching and nest ecology can become a major public health concern. If someone hands you a flea and asks you to examine it under the microscope, where do you start? First, recognize the basic flea body plan. Fleas are laterally compressed, meaning that they are flattened side to side, an adaptation that helps them move in between hairs. They are not dorsoventrally flattened like lice. They also have powerful hind legs. Fleas are tiny jumping machines. If insects held track and field events, fleas would be smug and unbearable at the award ceremony. Next, look for the combs. The genal comb sits on the head, while the pronotal comb lies on the posterior edge of the pronotum, the dorsal plate of the first thoracic segment. Genal simply means cheek, so the genal comb is literally the flea's cheek comb. These combs, properly called ctenidia, are not just decorative mustaches. Their backward-turning spines act like tiny barbs, helping the flea remain wedged between the host's hair when the host would very much prefer otherwise. They are also valuable identification clues. Tennicephalides species have both genal and pronotal combs. Pulex, Echidnaphaga, and Xenopsilla lack combs. So when you see both combs, Tennicephalides should rise quickly on your differential list. When combs are absent, slow down and consider the no-comb fleas, especially when clinical or public health contexts point in that direction. Identifying every flea to a species level is not part of the average veterinarian's daily work. However, it is important to recognize that different flea species exist and to understand the basic logic used to distinguish them. A complete identification key may consider head shape, the presence or absence of eyes, the position of the ocular bristle, the mesopleural rod, and several other structures. One of the first high-yield questions, however, is whether a genal or pronotal comb is present. This chart is only a reference. Flea diversity extends far beyond these examples, and some species may only possess one of the two combs. Host association provides useful context, but it does not prove identity. Many flea species can feed on more than one type of host. Finding a flea on a dog, therefore, does not necessarily make it a dog flea. Morphology, host association, and ecology must be considered together. Species identification may be particularly important during public health investigations. For example, identifying the fleas involved can help clarify vector and reservoir relationships during outbreaks. Outbreaks may include Yersinia pestis, the causative agent of plague, or Rickettsia typhi, the agent of flea-borne or murine typhus. Now that we know what a flea looks like, let's talk about what it does after reaching the host. Fleas begin feeding within seconds and can produce flea dirt very quickly. That means that even rapidly acting flea products and repellents may not prevent every flea from taking an initial blood meal. The goal is to kill the flea quickly and prevent the infestation from becoming established, not to create an invisible force field around the animal. Female fleas are impressive feeders. As they take a blood meal, their bodies can expand dramatically, much more dramatically than males. Fleas may consume roughly 15 times their body weight in blood each day. 75 Tennicephalides fleas consume 1 milliliter of blood a day. No wonder they can generate anemia. Tiny insect, enormous appetite, almost no respect for portion control. The clinical takeaway is this: seeing a flea bite does not necessarily mean that preventive failed. It may mean that the flea encountered a protected animal and is now living on borrowed time. Flea eggs fall into carpets, bedding, furniture, and cracks in the environment. Larvae hatch and they feed on organic debris, especially blood-rich feces produced by adult fleas. In other words, adult flea dirt becomes baby flea food. When adult insects provide resources and energy to their offspring, that is called parental investment. Thus, adult flea poop becomes baby flea food and promotes development. Parental investment, flea style. The larvae then grow and develop. When ready for their next stage, the larvae spin sticky silk cocoons that collect dust, hair, and carpet fibers. This camouflage helps protect the developing pupa and makes those cocoons very difficult to remove by vacuuming. That camouflage also helps protect them from one of their biggest predators, ants. To provide additional protection, their silk is even an ant repellent. Once they've developed this pupa, they will wait until carbon dioxide, warmth, vibration, or movement signals that a host is nearby. Humidity also matters. Fleas and their immature stages lose water quickly, so survival decreases when relative humidity falls below about 50%. Flea control requires patience. Treat the pet, manage the environment, and remember, the next wave may already be hiding in the carpet. That completes the flea strategy. Adults feed rapidly on the host, while most of the developing population waits in the environment. Lice use a very different approach. Now let's move on to lice. If fleas are jumpers, lice are clingers. Lice are wingless, flattened insects with claws built for grasping hairs or feathers. They are not built for athletic jumping. They're built for staying put. The practical split is chewing versus sucking. Chewing lice feed on skin debris, hair, feathers, scales, and surface material. They tend to cause pruritus, rough coat, feather damage, scaling, and poor thrift. Sucking lice pierce the skin and feed on blood. Heavy sucking louse infections can cause anemia, especially in young, debilitated, or heavily infested animals. This is where host specificity becomes your friend. Lice often behave like tiny little bouncers checking species ID at the door. Dog lice tend to be dog lice. Cat lice tend to be cat lice. Poultry lice tend to be poultry lice. Nature loves exceptions, but host species is usually one of your strongest clues with lice. Sucking lice reside in the suborder Anopleura. These blood feeders only affect mammals. Chewing lice span several different suborders. Ischnocera means slenderhorn, referring to their delicate antenna. Amblycera has blunt horns, thus look to the antenna to help differentiate. Rhynchopithecina is a specialized group, mainly elephant and warthog context. Wonderful if you are at a dinner party with louse taxonomists, not a core domestic animal louse. Always start with the host species, then look at morphology, feeding behavior, and affected body site. Now let's review major species affecting domestic animals. In dogs, the classic chewing louse is Trichodectes canis. The name means hair biter of the dog. Like the cat flea, this parasite can serve as an intermediate host for Dipylidium caninum, so it's not just a coat quality footnote. The suffix cola is important to remember. It means dweller. Thus, the cat louse, Felicola subrostratus, means the cat dweller, who is somewhat beaked or slightly snouted. The louse boasts a short, broad head and mandibulate mouth parts that kind of resemble a beak. Cat lice are uncommon in healthy, well-groomed cats, so when you see them, think about debilitation, poor grooming, long hair, feral status, or a shelter context. For ruminants, the common lice include Bovicola and Lingonathus species. In swine, Hematopinus suis is the big one and means blood-feeding pig louse. In poultry, think Menacanthus and feather damage, irritation, and production loss. Host species plus feeding style gets you far. Now we shift from taxonomy to the exam room. Start with the patient and the pattern. Is the animal itchy? Dorsal lumbar alopecia and dermatitis in a dog? Start looking for fleas and flea dirt. Flea dirt is digested blood in flea feces. One nice trick is the wet paper towel test. Flea dirt can turn reddish brown when moistened. That's not glamorous, but parasitology never promised glamour. It promised tiny clues with big consequences. Fleas can lead to life-threatening anemia in some cases. For lice, look for nits and adults on feathers and fur. For sucking lice, keep anemia on the radar. So when you see itchy skin, do not jump straight to a diagnosis. Ask host, site, debris, behavior, and parasite form. The patient is usually leaving you clues. Let's talk transmission and public health patterns. Fleas are the bigger vector and intermediate host story in veterinary medicine. They help transmit or help maintain several important organisms. Rickettsia typhi causes murine or endemic typhus, and flea vectors include Xenopsilla chiopus and Tenosiphalides felis. Rickettsia felis is associated with flea-borne spotted fever, or murine typhus-like disease, especially with cat fleas. Yersinia pestis, the plague agent, is classically associated with Xenopsilla chiopus and other rodent fleas. Bartonella henselae is linked to cat scratch disease with flea-associated transmission in cats. And Diploidium caninum uses fleas as intermediate hosts. That is why fleas are not just itchy punctuation marks. They are mobile ecological connectors. Lice are usually a different story. Most animal lice are highly host-specific and mainly cause irritation, pruritus, poor thrift, coat damage, feather damage, or anemia. But there are important exceptions. In pigs, Hematopoenis suis is associated with smallpox transmission. In humans, the body louse, Pediculus humanus corporis is the major disease vector louse linked to epidemic typhus, trench fever, and louse-borne relapsing fever. Historical note: trench fever even brushed against literature. Both A.A. Milne and J.R.R. Tolkien were sent home from World War I with trench fever. So in a strange way, body lice and Bartonella quintana sit in historical background of both the 100-acre wood and Middle-earth. Tiny vector, enormous literary side effects. So fleas are usually the bigger veterinary vector story. Lice are usually host-specific clingers, with a few high-stakes exceptions. Diagnosis starts with the pattern. What's the host? What's the behavior? Where in the body are the parasites or lesions? Are we seeing flea dirt, nits, adults, anemia, scaling, or feather damage? For fleas, flea combing is a high yield, especially over the dorsum, tail base, or caudomedial thighs. Look for adults and flea dirt. Remember that a patient can have severe flea allergy dermatitis even if you do not find a parade of adult fleas. The immune system can overreact to a bite even when the parasite count looks modest. Biology, as usual, is being dramatic. While isoxazolines for all furry four-legged family members can help resolve flea infestation, environmental control still helps. Vacuuming and washing bedding can reduce immature fleas in the environment. Due to pupil resistance, sometimes the full life cycle still needs to run the course to eliminate. This helps maintain the importance of veterinary professional recommendations for year-round flea and tick control. For lice, diagnosis often comes from seeing adults or nits attached to the hair or feathers. Host species is a major clue because lice are usually host-specific. Control is more host-focused. Treat affected animals and close contact animals. Clean equipment and bedding, reduce crowding, and improve nutrition and husbandry. Know your parasites and their life cycles so you may better treat and prevent. Let's pull the pattern together. Fleas jump, lice cling. Fleas have adults on the host, but immature stages develop off-host in the environment. Lice keep the whole life cycle on the host: eggs, nymphs, and adults. Fleas often leave flea dirt, allergic skin patterns, and public health or intermediate host stories. Lice often leave nits, rough coat, feather damage, anemia in sucking louse infestations, and strong host pattern clues. Exceptions matter. Xenopsilla chiopus is an important public health flea. Pediculus humanus corporis is the major human body louse vector. Hematopoenis suis is an important veterinary louse exception in pigs. If you remember nothing else, remember the clinical sorting system: movement, host, visible debris, feeding style, and life cycle. That is how fleas and lice become manageable instead of just a drawer full of itchy Latin. Thanks for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.